We are ready for the last session of the conference. The really hardcore devoted colleagues who are staying for the discussion and coming up with community standards for some things that are very important for us. And I'll start with something that Roman suggested. He took the initiative of suggesting a very important topic to discuss, which is how do we present our results as a community of how we quantify proteins in single cells to be most convincing for the broader community. That's a very important topic. There certainly has been a lot of skepticism, and some of it well-founded by people who understand why they're skeptical, and some of it just being generally skepticism born out of I didn't do it and I haven't done it in my lab. So I think it's a good topic that Roman is going to lead and discuss. Yeah, over there. We are going to break into groups to discuss that. And earlier today, I asked Jorgen to lead the discussion of data sharing and integrating different software that different groups develop into a common platform that is user-friendly and easy to use. And he's certainly somebody who has been incredibly successful from my perspective in doing this with MaxQuant, making it very user-friendly, which is well testified by its wide user base. So if you're interested in discussing this, Jorgen is your person. He's going to lead that discussion. And then here I have a couple more questions that I had suggested. One is what are the biggest marginal gains among all of the different opportunities that we discussed throughout the 2 days of the conference? What are the changes in our workflow that are likely to give us the largest improvement, either in throughput or the quality of the quantitation? And does anybody want to lead that discussion? I don't know. Well, and then we have another topic. Let's see. What are the benchmarks for quantitation that we can use? That's personally one of my favorites because I think it's incredibly important. This morning, John Yates said something that I think we should all learn from, that in the early days of analyzing mass spectra, there was a lot of good work happening and there was some not so good work happening, some sloppy identifications that instead of helping the field, contributed to eroding the confidence of the broader community and perhaps delayed the adoption of what was otherwise inevitable and very good change to the way mass spec data was analyzed. So I think we as a community should be very focused on establishing rigorous criteria so that we can ensure that our claims are well supported by data and they don't breed skepticism, but instead they help catalyze the fastest possible acceptance by the broader community, because I think there's a lot of expertise already existing within the mass spec community that can help accelerate the development of single cell techniques. Many of the people with that expertise don't believe that's possible. So the faster we can convince them, the faster they can help us to grow as a community, and I'm sure that there would be plenty of support from the various funding agencies and from the biology community. I have personally seen a lot of interest from the biology community. I'm sorry?
Is there anybody here with a lot of single-cell RNA-seq experience? Because that might be good to lead the discussion on how do they benchmark as far as...
I'm very familiar with that. So if nobody else is interested in leading that group, actually, I will lead it. So I'm very familiar with the analysis of single-cell RNA sequencing data. I've done plenty of that, and I certainly have strong statistical and math background, so I can be part of that discussion. But one thing that I can share with all of you just to begin with is that the first very significant and influential demonstrations of sequencing RNAs from single cells were not quantitative. They demonstrated detection of messenger RNAs. They did not quantify messenger RNAs. And based on the arguments that I presented early yesterday, even now, the best methods that have benefited from years of development and lots of resources detect most of the messenger RNAs. They cannot quantify them. Even if sample prep is perfect, even if sequencing is perfect, because of the inevitable sampling noise that is present, the relative error in those estimates is 50% or larger. And the community has realized that even though those relative errors are there, they can still use the data productively, and they can classify cells, maybe sometimes not so productively, maybe sometimes they exaggerate things and they form clusters out of cells that are really very similar, and they see clusters where clusters don't exist. But in general, we can learn from the example of using even noisy data in a statistically sound way, so that we can form conclusions that are nonetheless trustworthy enough. So talking about this, I will volunteer myself to lead the discussion on the benchmarks for single-cell mass spectrometry data. Anybody interested in leading discussion on the biggest marginal gains?
Appoint the volunteer.
I'm sorry?
Appoint the volunteer.
Appoint. Well,
I can facilitate it. I don't feel like I know the answer, but I'm happy to.
good. Okay, so Cherry is going to lead that discussion. All right, so let's break into groups. We are going to have about 30-minute discussion within the group, and then we are going to reconvene, and each group leader is going to succinctly summarize some of the important conclusions from the discussion. Jorgen, do you want to start? Yeah.
Yeah, I think we got cut off, so we could have probably discussed quite a bit more. And I think we found then two main points that we talked about and thought that important. And also one thing is, I think that can be translated into action items quite quickly. So actually one was, what we talked most about was actually QC. So that one maybe, as long as the community is still small, uses this opportunity and agrees on some QC standards. Because once we are 1,000 people, just think about general like bulk proteomics, they don't ever agree on anything basically, right? So now that we're only 20 or 30 people, we should really use that opportunity. And so, for example, you are already having QC scripts in your lab, right? And so that's of course then a good assumption that this is doing reasonable things. And maybe we can just use this as a starting point, right? And so one thing one would like to have then is probably some open place where these QC scripts are hosted and also visible to everybody. They can be discussed there if they should be changed, but there's only one QC script that basically everybody can run on their sample and then get answers about do I see single cells, is my coverage good enough, and so on. So that all these questions, these basic technological questions are answered. So then the question is, of course, in what kind of language should that be written? So I've learned you're using a mixture of MATLAB and R at the moment, is that correct? MATLAB and R? So the only issue with MATLAB is that you need a license, right? So it would be great if those could be converted into a mixture of R and Python, maybe, because then you don't need licenses for these, right? So that's what we can, so that's basically the suggestion, so that we have maybe a project in GitHub that everybody can see, that you convert the QC that you have now into a combination of Python and R, so that nobody needs licenses for these. Everybody can see these, discuss these, download these, and that this would be maybe one standard that we can, us 20 people can agree on now, basically, right? And then whoever joins the community, sees, oh there's already something, and will be very happy that there is already some standard, basically, right? So that's one thing we were thinking. So then we were also discussing about maybe some sort of a web server, or let's first start with the data sharing. So data sharing in bulk proteomics, I mean, we do have these repositories for these, like for example, Pride. So massive, I'm more familiar with Pride, I don't know that much about the other ones. But so that seems to be working, so my impression is that seems to be working quite well. So actually most people are actually depositing their bulk proteomics data to Pride. And as a means for just providing the raw data and distributing the raw data, I think this works very well, right? Unfortunately, if you go then one level higher, so even just being able to browse the MSMS spectra, so simple things like this is actually not working that well, because most of the submissions that are done to Pride are actually incomplete. So it's just the raw files itself, and then the results in some unstructured form. So often it's actually the max quant output, basically, right? Yes.
So Protein Prospector has a really good MS2 viewer.
Okay.
That you can upload, and I haven't done it in a while, but you can upload basically your search results, and it's clickable. So you can click on the peptide, it'll show you the MS2, and you can actually change the sequence to see if you put it. It's called MS Viewer. It was really great for like PTM reporting.
Yeah. I think so. Everybody has this isolate solution. I mean, you can also say Max Quant Viewer is great for looking at these projects, but there is no common standard that everybody agreed to, basically, right? And then Pride is like the best we have in terms of common standards. It's very good for distributing the raw data and making it available, but going beyond that, it's actually not really working well, I would say, in the community. And probably same. I don't know about Massive, probably same, I would think. But then one could just say, okay, then the basic thing of just providing the raw data, one could just use, I don't think we need an extra platform for this. This could be just the usual proteomics platform, so be it Pride or Massive now, right? So that at least the raw data is available. But then one wants to go maybe one step further, that there would be some sort of web server, some application that allows you to browse the data and also allows other users to browse your data. So that I think the time was not really enough to specify what that exactly should look like. But so not just identification quality, but going a bit further, that you can really browse through the quantitative data, right, and make your own impression of that. But I can already say now, to standardize something like this is a major issue. And if the community would be a little bit bigger, I would say it's impossible, because in proteomics it was not possible to establish something like this, right? And now we are at this point now, at least it looked like that everybody who is doing single-cell proteomics is actually using max quant, right? So I'm of course not the person to enforce that onto anybody, but so maybe to just get this development started, to have some kind of browser, web server to view the data, one could say, okay, now we just, we have the max quant output tables in mind and have something that works on this, but we should not get locked into this. So if certainly at some point we want to compare to Proteome Discoverer or any other software, so one should always have in mind that this server should also be able to work later, at least with Proteome Discoverer, maybe also with other platforms, right? But I think to make now a completely generic development effort where we say, okay, we don't know what software we're going to use, we now specify some crazy output formats in XML or whatever, this will not work, because we certainly don't have the manpower to work on these kind of generic concepts, and in the end there will be just disagreement and nobody. So like with the, well, I should probably not talk about, well, let's say like the standards that you have in bulk proteomics, actually nobody's using them basically, right? Because they were. No, I'm talking about like file formats and things like this, right? So like all these XML formats that people come up with, right? And then they just ignore that, vendors are never going to implement these and so on. And in the end, nobody's using them, right? So I think this is actually a big waste of time if we would start now with like what the generic file format for these kind of things looks like, right? So I think we should not do this. And just for practical purposes, I would say, okay, why don't we, okay, now we're still all using max_quant. Of course, I'm also interested in comparing with proteome discovery results, right? I'm the last person to try to log us into max_quant results. But one could just, this platform, one could just develop it for now on the max_quant output formats and then see how we develop it further, right? Did we have anything else? Yeah, I think that's it.
Anything about spectral libraries maybe as a resource?
Yeah, would be. But this, I think this is going to be very similar to bulk proteomics, right? So I don't think, probably we won't need special spectral libraries. Okay.
Wow, a lot of good ideas. And what is striking to me is that I see emerging themes in these different groups. We broke out into separate discussion groups with different topics, but one thing that comes through is the quality control, having some common standards. And this was one of the first things that Jorgen talked about and Cherry also mentioned. We spent a lot of time also discussing it in terms of benchmarking our data. And I have to confess that when I first thought of the benchmarking discussion group, I didn't think that we'll be discussing having the same shared standard among everybody, but we spent most of the time talking about that. We agreed that they will be very good if we can have some shared reagents that can be easily distributed even just to mass-spec facilities and mass-spec labs who don't have the ability to grow their own cells and sort them. And there is a technical challenge in terms of being able to do that, in terms of shipping frozen plates and of single cells and making sure that the single cells still stay where they're supposed to be. And then we also discussed that shipping cell lines that express fluorescent proteins that then different investigators can use to sort and use for single cell prep standards is something that may offer a compromise between having an easily distributable reagent and lowering the energy barrier a little bit, though not as much as providing samples that are ready for prep. We, I think, had unanimous agreement that fluorescent proteins are a good way to benchmark our measurements, both in a qualitative way, that if some cells express M-cherry, it should be present there, but not in the cells that don't express it, and in a more quantitative way, where we can use the fluorescence of these proteins to derive relative abundances and use these to benchmark to mass spectrometry. There is also another emerging theme that I can see among the different discussion groups is the realization that while the community is still relatively small, introducing many of these things will be easier than later on as the community grows. And a good piece of advice from John Yates that we are on the right path having started this conference, because one way to get these standards is to get interest from vendors and companies in standardizing them and providing them. And that will be easier if we have a shared meeting and a growing community. So we've made the first step, and I think it has been a very good step, certainly very, very enjoyable meeting for me. And I'm very much looking forward to making the second and the third step and staying in touch, not just seeing you next year, but trying to have more frequent interactions and trying to have a follow-up on some of these very good ideas that we came up with. I think that there is a very good momentum. It's just the beginning, and having momentum at the beginning is promising. It's also something that we can lose. I don't think that we have yet crossed the threshold where we are unstoppable. Hopefully we'll do that, but there is still need for a concerted effort from all of us to be moving in the right direction, demonstrating the feasibility of single-cell proteomics by mass spectrometry, its throughput, and also making it much, much easier for the broader community to adopt, because as Roman mentioned in his talk, if something is working in a couple of labs, Maybe it's true, maybe it's just good luck. It is when it is adopted by dozens of different laboratories that it begins to be a standard routine procedure that everybody believes is possible. And I hope that in the not too distant future, we'll be at that point and we'll get there with our common efforts. So thank you so much for making this meeting possible, coming here and sharing exciting results, thoughts, and ideas, getting to know each other. It's been very, very fruitful and wonderful interaction for me, and I look forward to seeing how it's going to progress. Thank you again.